presence and structure of the plasma membrane, but the fluid mosaic model is proposed by the Singer-Nicholson. It is the most acceptable type of the model of plasma membrane. It is reported in 1902. Overton proposed a basic model for the transport of the small neutral solutes. It can be experimental numerous cells, both from the plants and animals, conducting these special osmotic properties of living protoplast. Overdone, the postulate in the plasma membrane is composed of the thin layer of the liquid. In 1926, Groter and Gradle conducted the experiment, the homolyzed RBC, and conducted that the RBCs were covered by two layers of the lipid molecules over the enhanced the cell surface. In 1931, so Daniel and Harry studies the surface tension of the cells, generally surface tension of the water-oil interaction interface around the 0.01 to 0.15 neutron meter. For other end surface of the tension of the cells, this low surface of the tension for cell is attributed to the presence of the proteins in the plasma membranes. The following are molecular models of plasma membrane proposed by the various biologists. So this is also first model, that is a sandwich model in 1935. So Davison and Daniel proposed the sandwich model of the trilamellar models. Then outer and inner middle, the three type of the layers are there: extracellular and also intracellular, between hydrophilic aids and hydrophobic aids. That is also lipid, phospholipid bilayer. Then also it is also outer globular protein and inner globular protein. This, so it also predicted the thickness. Then so next, sir, next. Then extracellular medium, outer proteins and inner protein. That is a unity membrane model for of the plasma membrane. This hydrophilic, hydrophobic. These two type of the lipids layer. Then outside protein, inside protein. Then three layers are there. Unity membrane is also model. Later in 1959, Robertson proposed the unity membrane hypothesis, which states that all cellular membranes have identical membrane structure. So they named this identical name structure is a unity membrane. The unity membrane is a second type of the model. According to this model, unity membrane consists of bimolecular lipids. The lipid layer is also two. It can be packed in the between the outer and inner layer of the protein. The protein is outer layer, inner protein. Then lipids are also got two. The two is also come in one layer. And outer protein, inner protein, between lipids. Then trilamellar model-like structures. Next. So this is also fluid mosaic model. This is also third type of the model, which can, the fluid mosaic is a, it can finally 1972, Singer and Nicholson proposed the well-accepted fluid mosaic model. As per this model, both lipids and proteins are distributed in a kind of mosaic arrangement. Mosaic means ice. The ice is when put into the water, ice is floating. The type, the phospholipids are float on the water surface. The total water is also lipid-like structures. The biological membranes are a fluid structures, which lipids and proteins can move. In other words, the proteins are embedded in the lipids bilayer in such a way that the proteins are float in lipid sea. The surface of the lipid layer is interrupted by randomly distributed protein molecules. These proteins are may either attach to the polar surface of the lipid or partially penetrate the lipid bilayer. Some proteins are also found in the be associated with the sugar chains of the glycoproteins. So next. This is also the fourth type is also micellar model of plasma membrane in 1963. So Hillary and Ahman suggested the biological membranes which can be lamellar pattern. As per them, the plasma membrane is a mosaic of the globular substances, subunits referred to as a micelles that are densely packed with a central core of lipids. As lipid micelles and spontaneously link, they are probably building blocks for membranes. The protein components of the membrane, this model can establish the monolayer or either side of the plane of the lipids. It is suggested the gaps between the globular micelles form water-filled spores, which are partially lined by a polar groups of the micelles and partially by a polar groups of associated proteins or molecules. Next. So this is also prokaryotic and eukaryotic plasma membrane differentiations. In prokaryotic and the plant's plasma membrane is the inner layer protection in the presence of the outer cell walls. Then also eukaryotic, in eukaryotic, the plasma membrane is the outer layer of the protection. That is also most differentiation of the prokaryotic and eukaryotic. First differentiation. Second one, the cell wall as a pores and is not very selective about the passage of molecules. Here, the plasma membrane is responsible for selective permeability of the molecules in prokaryotic cells. In the absence of the cell wall in eukaryotic cells, plasma membrane is completely responsible for the selective permeability of the molecules in animals, but plants permeables. Then third one, prokaryotic plasma membrane is a phospholipids bilayer with embedded in the proteins. Then eukaryotic, it can plasma membrane is a phospholipids bilayer with embedded proteins and carbohydrate. Just differentiation between the prokaryotic and eukaryotic. The prokaryotic plasma membrane has no sterols, but eukaryotic plasma membranes has a sterols like cholesterol in the hydrophobic cores. Then the prokaryotic may have multiple plasma membranes. In the eukaryotic, they have only one plasma membranes are there. Then prokaryotic, in the absence of the sterols, the plasma membrane is a prokaryotic in the less rigid and less stable. Then last of the eukaryotic is the presence of the sterols gives more rigidity and stability of the membranes. So next, this is all type of the models of the differentiations. Model name proposed by year, structural description, key features and limitations. Then first one, lipid mod by and a bilayer model over 1902. Garter and also Grader suggested bilayer lipids. It is a hydrophilic aids and the outside did not expand the membranes permeably and functionally. It is also first. Second one, so Davis and Daniel model. It proposed by the James Daniel Ungu Davison. It can also got a sandwich model of lipid bilayer covered with proteins layer. Then key function is a then also lipid bilayer core protein coat outer surface. First molecular level of model members. Then limitation state of the protein layer, so it could not be explained the selective permeability and a protein movement. Then third point is a unity membrane model. Again, proposed by the David Robertson. Is also structurally is also trilamellar, 3 layers structures observed in the electron macrography. Then keywords also feature is a common structural for all membranes. Then second, dense protect protein units and light lipid layers. In the limitation is also got a assumed to the informed also thickness. It can be failed to the explain of the systems. Then last model, fourth one is also accepted in all scientists. It is also best model of the plasma membrane. That is fourth one, fluid mosaic model. H. Singer in G.L. Nicholson, 1972. Dynamic model with proteins embedded in fluid phospholipid bilayer
Sunday
means
ఇంటికి ఎక్కడ
phospholipid
ఇంటిని ఎక్కడ
bilayer
ఇంటిని ముక్కలు
with embedded proteins. Then protein move, also called a expel fluidity, then also limitation, most accepted and updated model, dynamic account for diversity in the protein movement. That is a fourth model. It's a very excellent model.
కొన్ని నీళ్ళని పంపి డబ్బాలు
Next.
కూసప్ప పెద్ద మామ
Next 24, sir. Hello. So this is also called the major parts of the year is complete the total topics of the plasma membrane sheet filling the blanks. The fluid mosaic model was proposed by dash and dash. It is important. Synchronicals. The plasma membrane primarily composed of the phospholipids and proteins are easy. Then last, the third one is the hydrophilic heads of phospholipids face aqueous environment. Then also called hydrophobic tails are phospholipids face inwards. That is only just a differentiation. Then also the hydrophilic is a phospholipid face aqueous and hydrophobic means inward. That is different. The first suggestion of the lipid is also then the first suggestion of the lipid-based membrane came from the Overton in the year 1902. Gordon and Gordon proposed the lipid bilayer model in 1925. And next, seventh one. Next. Then Davis and Daniel model is also known as the sandwich model. The Davis and Daniel model, which can be in lipid layer and sandwich layer, that is important of the next part, sir.